Hello students, welcome to e-learning session in microbial technology. Today in this session, we learn about microbial sweeteners. Sugar is sweet in taste, which is present in cane and beet and is consumed most commonly as sweetener. Other sweetener example is fructose, which is present in fruit and honey. It is cheaper than sucrose, and this fructose is more suitable for diabetics. The other examples of sweeteners are aspartame, saccharin, Tamartin, HFCS, etc. These sugar substitutes can be classified into two groups. One is nutritive or caloric sweetener. The second one is non-nutritive or non-caloric sweeteners. So these nutritive sweeteners are metabolized to provide energy, whereas non-nutritive sweeteners do not provide energy. So these sweeteners are substances of low energy value that provide sweet taste, and these sweeteners have been used as sugar substitutes in many sweet tasting products. Although there is great interest in and demand for these substances, their use or their application is not always easy. Now we'll see few examples of sweeteners. First, we'll take up the sweetener, that is aspartame. Aspartame is an artificial non-saccharide sweetener which is 200 times sweeter than sucrose and is commonly used as a sugar substitute in foods and beverages. Chemically, it is a methyl ester of the aspartic acid or phenylalanine dipeptide. With the trade names, it will be sold in the market by names like NutraSweet, Equal, and Canderel, etc. So these are the commercially available aspartame in the market. So this aspartame was first made in 1965 and it is approved for use in food products by the US Food and Drug Administration, that is FDA, in 1981. This aspartame is produced by coupling microbial fermentation and synthesis. Phenylalanine and aspartic acid are produced by microbial fermentation, and this phenylalanine is reacted with methanol to form methyl ester. Phenylalanine will react with the methanol to form the methyl ester. Then the modified amino acids are mixed in a reaction tank to get aspartame intermediates. It is further treated with acetic acid. Later, it is purified and crystallized, and this will be powdered to produce the sweetener, that is aspartame. In addition to this microbial fermentation, many methods for direct enzymatic synthesis and chemical synthesis are also reported for the synthesis of aspartame. So this is 200 times sweeter than sucrose, and it is a methyl ester of aspartic acid and phenylalanine dipeptide. peptide. So this can be produced by the combination of microbial fermentation and the synthesis. Apart from this, aspartame can also be produced by direct enzymatic synthesis, and chemical synthesis are also available for the synthesis of aspartame. Next, we'll move on to the next sweetener, that is Tamartin. Tamartin is a flavor modifying and sweet tasting protein which consists of a single polypeptide chain consisting of 207 amino acid. A single polypeptide of 207 amino acid. It is derived from the fruit observed in West African tropical plant that is called Tomatococcus danieli. This natural sweetener was first noticed in 1840 by a British surgeon in Africa who found the native people using this plant fruit to sweeten the sour wine and food. In the present day, it is very popular throughout Europe and Japan. In It is used in preparation of foods and drinks. Commercially also, it is available in the market under the trade names like San Sweet T100 and Talin. These are the trade names for the commercially available Tamartin. This Tamartin is soluble in water and aqueous alcohol and is stable in freeze-dried form. And also it is pH and heat stable. And it is synergistic when it is mixed with other low-calorie sweeteners. In addition to this, it masks the bitterness and increases the efficiency of flavor enhancers such as monosodium glutamate, that is MSG. That is regarding the Tamartin. Next, we will move on to the another sweetener, that is high fructose corn syrup. In short form, it will be pronounced as HFCS. It's also known as glucose fructose isoglucose and glucose galactose syrup. Glucose galactose syrup. It's a sweetener made from corn starch. It's a sweetener made from corn starch. As in the production of conventional corn syrup, the starch is broken down into glucose by enzymes. To prepare HFCS, the corn syrup is further processed by the another enzyme, that is D-xylose isomerase, to convert the part of the glucose, proportion of the glucose into fructose. So this HFCS, high fructose corn syrup, was first marketed in early 1970s by Clinton Corn Processing Company together with the Japanese Agency of Industrial Science and Technology, where the enzyme was discovered in 1965. As a sweetener, this HFCS is compared to the granulated sugar, but manufacturing advantages of HFCS over sugar include that it is easier to handle and cheaper. There are many commercially available HFCS. They are named like HFCS 42 and HFCS 55. So this refer to the dry weight of the fructose compositions of about 42% and 55% respectively. So the remaining proportion will be the glucose. This HFCS 42 is mainly used for processed foods and other breakfast cereals. Whereas this HFCS 55 is used mostly for the production of soft drinks. So this HFCS will be produced by using the gelatinized starch slurry. What they'll do is heat the gelatinized starch slurry of about 30 to 35% and it will be liquefied. And later it will be dextrinized by the bacterial thermostable alpha amylase in two-stage reaction. Initially, it will be incubated at 104 to 107 degrees centigrade for 5 to 8 minutes. It results in the formation of dextrose equivalents, hydrolysate. After lowering the temperature, it will be passed through a column of immobilized isomerase with a residence time of zero point 0.5 to 4 hours. Then it gives the glucose will be converted into fructose. Like this, HFCS will be prepared. In addition to that, there are other sweeteners also like fructose sweet. It's a mixed product of aspartame and acesulfame. Along with that, selected fruit concentrates will be mixed in the fructose sweet. Even this will be sweet in taste. So in this session, we learned the importance of sweeteners, how they are produced, and their trade names in the market, how they are sold in the market commercially. So these microbial sweeteners are having the advantages over the chemically synthesized sweeteners. I hope you understood this session. Thank you.